Noswaith dda, chroeso I chi gyd. Colin Redden dwi, Ysgol Feddygaeth Caerdydd. Well, good evening, everybody, and welcome to today's event. I'm Colin Redden, the Vice-Chancellor of Cardiff University. Now, this is a subject we have today, which I know will have touched many, if not perhaps all, of us here on the call this evening, either directly or indirectly, because Cancer Research UK estimate that over 374,000 people in the UK are diagnosed with cancer every year. And of course, all those people are likely to have others who are close to them, relations, loved ones who are also impacted by this. Of course, research has made enormous progress over the years, and particularly in recent years, as our techniques in terms of genetics and understanding of mechanisms is improving, and that is helping. Cancer survival itself is then therefore improving. Now, 50% of people survive their cancer for 5 years or more, so there's a clear improvement there, but there's always much more to do. We're a long way from really being able to fully understand and resolve this problem. And that's why for us at the university, cancer research is one of our principal biomedical themes. And what we do is to bring together expertise in cancer genetics and tumor development right through to new drug discovery and clinical trials. So it's a whole spectrum from the fundamental research right through to clinical trials for therapy, various forms of therapy. We're also a partner in the Wales Cancer Biobank, and what that does, it asks patients in Wales who have known or suspected cancer to consent to donate samples and data that we can then use in cancer, and others actually can use in cancer-related research. And we have over 15,000 consenting patients that have made that resource into the fantastic resource that it is today. It's one of the UK's largest and longest established biobanks, and it's an award-winning asset, actually, that makes our research and future treatments possible. So you've been invited here today as part of the Cardiff community, and you include alumni, staff, donors, students, I'm sure, and friends who are supporting the learning and research of the university. We are really grateful for your support in this area, because it wouldn't be possible without your generosity and that of the public. So thank you, diolch yn fawr, for that. So tonight's event is specific about a particular area, which is our research in cancer immunotherapy. Now, I'm not going to try and explain what that is. That's the job of our experts this evening. And in a moment, we'll hear from them. First of all, Professor Awen Gallimore. Now, Awen actually leads our research in cancer overall for the university, but also particularly in immunotherapy research. So as well as setting the overall strategy and priorities, Awen specialises in the area that we're talking about today. And she's accompanied by Lorenzo Capitani, who's a PhD student, also in the School of Medicine. And Lorenzo is investigating ways of making the immune system work as well as it can, helping the T cells you might have heard of in relation to COVID, to help the T cells element of the immune system fight cancer more effectively. We'll hear from Awen and Lorenzo in a moment. And after they've given their presentation, there'll be plenty of time for questions and answers. We have received some in advance, but if you've got any to ask, just put them in the Q&A box at the bottom of the screen, and we'll try to answer as many as time allows. Now, we can't always get around to them, but if you have put a question either in advance or in the box at the bottom of the screen, we will get around to answering them, and we'll get in contact afterwards. So everybody will get a kind of list of the questions and answers subsequently if we don't have time to answer them during the event itself. So that brings us now to the main part, which is the presentation. So I'm going to hand over to Awen. And Awen, you're welcome. Please do start your presentation.
Thank you very much, Colin, for the introduction. So I'll go and go to my slides, and once I have shared the screen properly with you, great. I shall tell you a little bit more about myself. So I am indeed an immunologist, and the main focus of my work is to try and find out how we can persuade the immune system to recognise and to kill cancer. And so this is an area that you probably know has generated and continues to generate a huge amount of interest and excitement as a new type of cancer therapy. So before going into those therapies, I first of all, I'm going to try and change my slides, yes, tell you a little bit about the immune system, because it's important so that we can understand how these new immunotherapies actually work. So the system has actually evolved to fight infections. So it's there to protect us against bacteria, fungi, viruses, different types of microorganisms. And it has to do this in a kind of balanced way. So it has to make sure that it leaves uninfected tissue alone. And this is very important because it protects us, therefore, against other diseases like autoimmune disease or inflammatory disease. So what happens then when the immune system sees a bug is that it responds to that. So this is a white cell here that has put on its surface a go signal. So that's what the little green dot represents. And by doing this, it activates more immune cells, more white cells. It recruits them to the site of where the infection is, and it clears the infection, and then the immune response wanes. The trouble is the good thing is when the immune system sees normal uninfected tissue, it will actually respond to this by putting up a so-called stop signal. And the problem, of course, with cancer is that it very much resembles, as far as the immune system is concerned, normal uninfected tissue. So it will respond to it by putting up these stop signals. And so the immune system is basically instructed to leave this tissue alone. But what we have learned, of course, is a lot about the nature of these stop and go signals. These are so-called immune checkpoints, and by understanding their molecular identity, what researchers have managed to do is generate antibodies. So these are these little Y-shaped, upside down Y shapes on the top of the screen that bind to these stop signals, stop them from working, and allow the immune cells to become activated instead in response to the cancer. And by using these antibodies, we've seen very significant breakthroughs in immunotherapy, which has enabled, as I've written here, the cure of previously incurable cancers, most notably melanoma, but now there are other cancers being treated with these antibodies, such as renal cell cancers, non-small cell lung cancers, and urothelial cancers. And this list is expanding. But of course, there's still a long way to go, and we know there are lots of current limitations. They work only in some patients. They're successful in certain cancers only. And there are always stories, such as you're seeing on the slide here, of how immunotherapy has made large tumours disappear. But again, this is still something that happens in the minority of cases. And of course, these immunotherapies are also toxic, because by unleashing immune responses that recognise ostensibly uninfected tissue, they're also recognizing normal, uninfected, non-cancerous tissue, therefore causing some toxic side effects. The good news is, though, that there are many, many opportunities, and there's huge scope for improvement. And I'm going to tell you a bit more about that now, and specifically a little bit more about our Cardiff research interests. So one of those is in a new stop signal. So it's not just the ones that, we already have drugs in the clinic to target that are at our disposal for exploitation. We're interested in a molecule called LAG3. This is a stop signal that we see on white cells which are present inside cancers, and Lorenzo will tell you a little bit more about this. And we're now starting to understand that if we block LAG3, we can then convert this stop signal into a go signal, and that this has got some significant anti-tumor potential that we're trying to harness right now. As well as the stop signals, there's another type of immune cell that my laboratory has been working on for over 20 years. It's a very interesting cell type called a regulatory T cell. And so this regulatory T cell is another form of white cell. but it's different to the aggressors, the one that go around killing infected cells or cancer cells should they get the chance. The regulator is actually there, again, to keep the immune system in check, to make sure that it gets activated at the right time, but that it then stops being activated when the danger has gone. So cancer is another situation whereby we see regulatory T cells when we don't want them. So for some reason, regulatory T cells become highly enriched inside cancers. We see this all the time. And what they do there is create a sort of immune suppressed microenvironment, thereby meaning that those immune cells that are there can't actually do their job properly. So of course, the challenge then becomes, how can we target these regulatory cells and get rid of them so that we can unleash a better immune response? So we have tested several drugs in order to look at their ability to do this. And one of those is called low dose of a chemotherapy drug, actually, called cyclophosphamide, which you use at much lower doses than you would for chemotherapy. And we found that this drug can actually target these immune regulators, boosting anti-cancer immune responses in patients. And what I'm showing you here is the results of, some results from a trial that was actually completed in Cardiff from our lab a couple of years ago, where we tested the hypothesis that cyclophosphamide could deplete regulatory T cells in patients with inoperable metastatic colorectal cancer. So this is very advanced disease, but we did see significant immune effects, and we also saw better progression-free survival in those patients who responded well to this therapy. And whilst, this study, because it was an advanced disease, was never expected to bring great benefit to patients because their disease is too advanced at this stage, these findings have now allowed us to get funding for an 800 patient study, which will start in Cardiff later this year. And this is a study that will be giving this same drug to patients with much earlier stage disease. So patients who have had their cancers surgically removed and who hope their cancers won't recur. And our hypothesis is that a brief intervention with this drug will prevent recurrence, at least in some patients. So we're also looking at targeting regulators in the context of triple negative breast cancer. This represents 10 to 20% of breast cancers, and it is an aggressive type. You see limited response to current therapies, so there is a need for something better and different. And we have some evidence that regulators are again enriched in the context of this cancer, and that targeting them could well unleash immune responses that keep the cancer under control. So I'll just show you some data from our lab using a sort of model system where we're looking at tumours growing in volume with respect to time. And you can see what happens if you give no treatment, the cancers grow quickly. If you use, here we're using a new drug, it's actually called a PI3 kinase delta inhibitor. I can share more details of that if you would like them. This specifically targets these immune regulators. And you can see there that the cancers grow more slowly in all cases, and in some cases don't grow at all. And what we actually saw was that in the case where the drug didn't work brilliantly by itself, so you're still seeing tumour outgrowth despite the presence of the drug, that the immune cells in those cancer contexts were now putting this stop signal, LAG3, on their surface. So we then gave targeted LAG3 using a LAG3 antibody, but saw actually no effect. In this case, the cancers continue to grow. But the very interesting thing is that when you use these drugs in combination, so you're targeting both the regulators, and then you're seeing that by using a drug to target the regulators, you're driving expression of this stop signal in some of the immune cells. If you then come back with a drug to target LAG3, you see, obviously, as it's very clear to see here, a far better anti-cancer effect. So that is the kind of work that we are doing. That's obviously a very small, short snapshot of the kinds of things that are going on. But I think it's enough to perhaps make the point that cancer immunotherapy is both significant breakthrough and untapped potential. There's so much more that can be done. And combination treatment and how to put them together. And these sorts of approaches, of course, require the integrated is different. Colin talked about the genomics that's going on. So it's very, very important to tailor treatments to individuals. And I'm also very happy to say that our research labs in Cardiff don't work in isolation. We've set up a joint research office with the University Hospital of Wales and also now with Felindre Cancer Hospital. So we have a tripartite Cardiff Cancer Research Hub, which, and the intention, of course, of this hub is to both harmonise all the regulatory issues so that we can work faster, but also so that we can join forces, so that we can have academics and clinicians and clinical academics working together with our partners such as the Wales Cancer Bank and the Wales Cancer Research Centre. And the whole point of that then is to accelerate translational and clinical research across the university, so we can bring our own discoveries to patient benefit faster. And of course, I'd also should at this stage thank our funders, in particular Cancer Research Wales and Cancer Research UK, who are significant funders both of our lab and the wider environment. My email address is here, so if there are questions that we run out of time for answering, I'm very happy for anybody to email me, and I'll hand over now to Lorenzo. And I shall stop sharing. Sorry, Lorenzo. There.
Hello, everyone. Thank you, Colin, for the introduction earlier, and thank you, everyone, for taking time out of your busy day for being here. I'm just sharing my screen now. Okay, there we go. So my background is also in immunology, and I'm a PhD student in the laboratory of Professor Awen Gallimore. And today I hope to talk to you about this stop signal that Awen talked about, called LAG3, and its potential as a novel target in cancer immunotherapy. So this is a little Star Wars reference in case there's any Star Wars audiences in the any Star Wars fans in the audience. And it's that immunotherapy, as Awen mentioned, is thought to bring a new hope to cancer treatment by awakening, quote unquote, the force within. So reinvigorating the immune response to better enable it to recognize and to fight and destroy cancer cells. And as Awen mentioned, in a variety of cancers, this has been very successful. For example, melanoma, allowing the treatment of previously untreatable cancers. However, the story isn't so successful in all cancers. And one of these cancers is colorectal cancer. So this is cancer of the colon and rectum. And in some clinical trials that have tested currently existing immunotherapies in colorectal cancer, 0% of patients show a positive response to this. Why is this a problem? Why should we be concerned with this? Well, this is because colorectal cancer has a significant health impact globally and is the 4th most deadly cancer, amounting for a total of approximately 900,000 deaths annually. And this is a number that is increasing. Why is that? This is because colorectal cancer is a disease associated with a quote unquote Western lifestyle. And you can see here by the world map. Here we have the world map, and each country is given a shade of blue. The darker the shade of blue, the higher the rate of colorectal cancer in each country. And you can see that the darker blue countries are associated, in more developed countries, Europe and North America. And this is because colorectal cancer is associated with a wide range of largely modifiable environmental lifestyle factors that we associate with a Western lifestyle. So, increased alcohol intake, smoking, reduced physical activity, intake of processed meat, reduced intake of vegetables, and obesity. So, as the world becomes more developed, undertaking a Western lifestyle, it's likely that the incidence of colorectal cancer is increasing. So it is a problem that, currently existing immunotherapies are inadequate to help us fight colorectal cancer. So this is why there is a need to develop new immunotherapies that are maybe better targeted to colorectal cancer. And this is where LAG3 comes in. LAG3 stands for lymphocyte activation gene 3, in case you're interested, but we commonly just refer to it as LAG3. And this is a three-dimensional diagrammatic representation of LAG3, or an estimation of what it looks like. And the interest of our lab in LAG3 is summarized from this graph taken from a study performed by Martin Skur in our lab a couple of years ago. So the vertical axis of this graph represents the percentage of white blood cells that have LAG3 on their surface, that have this stop signal LAG3 on their surface. And Martin Skur in the lab assessed the amount of LAG3 on the surface of white blood cells coming from different samples of white blood cells from different portions of patient tumors, of course, with patient consent. And you can see that white blood cells coming from the diseased colon, so that is a portion of the colon that actually had the colorectal tumor, had a higher percentage of LAG3 when compared to white blood cells coming from healthy colon or white blood cells coming from the blood. So this suggested to us that potentially LAG3 might be an important stop signal that is driving a weaker, more dampened, immunosuppressed immune response in colorectal cancer. So some of the work I have been doing is further trying to establish LAG3 as a potentially interesting target in colorectal cancer. And I have been doing this both by doing computer-based data analytics research, what we call bioinformatics, and also through carrying out research in the lab. I'm going to be giving you an example of both kinds of research. So in the bioinformatics aspect of my research, I've been trying to investigate whether we can actually associate LAG3 with a better or worse outcome in colorectal cancer. And this is summarized in this graph over here. So the x-axis is just time in days, and the vertical axis is the chance of a patient surviving over time. And this data comes from an online database of patients that have colorectal cancer from the NIH Institute in the USA. And what you can see here in orange is that patients that have higher amounts of LAG3 in their colorectal cancer sample have a reduced chance of survival over time when compared to patients that have lower levels of LAG3 in their cancer. So this again sort of suggests to us that LAG3 might be an interesting stop signal that is playing an important role in weakening the immune response in colorectal cancer. I've also been, doing more direct investigations of associating LAG3 with a weaker suppressed immune response. And this is shown in this graph over here, which I appreciate might be, a lot to take in, but I'm going to walk you through how we read one of these graphs. So this graph represents genetic information from approximately 500 patients with colorectal cancer. So each patient is represented by a very thin line over here. So each thin sort of column in this graph represents an individual patient. And then each row in this map represents a different gene group. And the amount of each gene group in each patient is represented by the color of each row. So the darker red rows show more enrichment in those individual patients. So whether a patient had lots of LAG3 in their tumor or little LAG3 is represented by the color up here. So you can see that the individual patients that had lots of LAG3 in their tumor are grouped up here. And what I found was that individuals that had lots of LAG3 in their colorectal tumor sample had higher amounts of gene groups that we associate with immune suppression, with a weaker immune response. And that's shown over here by the enrichment of a dark red color in the rows representing PD1 signaling, which, as Awen mentioned, is another one of these stop signals, and other stop signals such as interleukin 35 over here, and interleukin 10 over here. So again, this provides evidence to us that LAG3 is associated with immune suppression, at least in the context of colorectal cancer. Because of this, we are now interested in studying immune suppression, but immune suppression is quite a complex affair. And it's quite difficult to model and replicate immune suppression in a petri dish. So some of the in a petri dish. So some of the work that we have been doing is developing new approaches to allow us to study immune suppression in a dish. And I'm going to describe an example of this to you. And this is called, these are called organoids. So what we do is we are very lucky here to have connections with the University Hospital of Wales, where of course a lot of patients are treated for colorectal cancer. So we come into touch with patients, and patients that are willing and happy to contribute towards our research, that consent to helping us out. When these individual patients that consent to helping us out are undergoing a resection of their tumor, so that means when their tumor is being surgically removed, we get small samples of the tumor from the patient, and then we can grow the tumor outside of the patient into what are called tumor tissue organoids. So these are three-dimensional structures which under the microscope look a little bit like this. And they accurately, or more accurately, represent the three-dimensional structure that they are derived from. So in our context, these are organoids that replicate the colorectal tumor. And again, when we obtain patient consent, this is what organoids look like at a higher magnification. When we obtain patient consent, we also are able to obtain white blood cells from individual patients. And our aim is to use these sort of model approach where we have a three-dimensional structure that represents the tumor of the patient. And we also have patient-derived white blood cells to study how and when we should target LAG3. So one of these in-a-dish systems I've been using has helped us, again, further support the idea that LAG3 reduces white blood cell function. And this is summarized in this graph here, which, again, I appreciate might be a lot to take in, but I'll walk you through it. So we have two axes on this graph. The horizontal axis represents the amount of LAG3 on the surface of a white blood cell, and the vertical axis represents the amount of PD1 on the surface of a white blood cell, both of which, as Awen mentioned, are these stop signals. Then function of white blood cells under different conditions is represented by the size of each dot, by the size of each bubble, right? So the larger the dot, the more white blood cell function we see in a given context. And so some of the experiments in a dish that I have been doing have shown that actually as the amount of LAG3 and PD1 on the surface of a white blood cell is low down here, the bubbles are quite large. And this means that the less LAG3 and the less PD1 you have on the surface of a white blood cell, the more functional the white blood cell is. But as the amount of LAG3 and PD1 on the surface of a white blood cell increases, they become less functional. So this, alongside the previous bioinformatic data that I have shown you, suggests to us that LAG3 might be an interesting target for improving the immune response in patients with colorectal cancer. And the idea is simply that by targeting LAG3, either through using one of these antibodies that Awen mentioned earlier to block LAG3 function, we might be, this is, a very tired and immune suppressed immune cell represented here. Using these antibodies, we might be able to target LAG3, reinvigorate the immune response to, give it that extra boost that it needs to get rid of the cancer cells. And we hope that the research we are doing will be a foundation for future immunotherapies. And we're not the only ones studying LAG3. LAG3 is a very hot topic now. This is data from a recently published clinical trial in the New England Journal of Medicine, comparing how effective a PD-1 blocking therapy on its own, here in green, and a therapy that blocked both PD-1 and LAG3 in blue, yw'r. And this study found a modest improvement in the progression-free survival of patients. These were patients that had melanoma. A modest improvement in progression-free survival in individuals that were given both PD-1 and LAG3. But you can see that the improvement is modest, and there's still a long way to go if we are to understand how we can maximize the benefit of LAG3 targeting immunotherapies, be it, as I mentioned, through the direct blocking of LAG3 function, be it through the maybe adding a toxin on these antibodies that target LAG3 and directing toxins to LAG3 expressing cells to kill them, or through a combination of a range of treatments, as Awen mentioned, blocking LAG3 and other targets, for example, PD-1. So hopefully the research that we are doing will enable us to improve how we can target LAG3 in colorectal cancer and beyond. So I hope you found this sort of overview of the research that we're doing interesting, and thank you for your attention.
Well, thank you very much, Lorenzo and Awen. So we've actually got about a good quarter of an hour for questions, which is great, and we've already had some in. I noticed that, Lorenzo, at one point there you mentioned that understanding the immune system is a pretty complex issue. I think that's something of an understatement, as you've very much demonstrated there. There are huge complexities here. And I'll just reiterate that we will send a link to the recording of the whole thing. So if you have, I understand some people have some difficulties with reception, but you'll be able to get it in clear. And if you just need to go back over it, because there's a lot there, so you will get that. But there's a couple of questions around the same sort of theme. And this is to do really with the complexity, I suppose, but which cancers seem to be responsive to immunotherapy and which ones are unresponsive, and why is this? And I guess that could be, is it the site? Is it the type of cancer? Is it solid or not? Or even at the same site, you could have different types. So yeah, who wants to have a go at that one? Which ones are responsive and which less so?
I can try if you, Lorenzo, yeah. Okay, so the cancers that appear at the moment to respond best to immunotherapies are those with the highest number of mutations in them. Mm. So, cancers, of course, emerge as a result of an accumulation of mutations that allow those cells to grow uncontrollably. But what also happens when you have many, many mutations is that you end up with altered proteins, because those mutations are encoding for slightly different proteins. And then it's easier actually for the immune system to see that cell as foreign. So in the case of melanoma, for example, that's really been the trailblazer for immunotherapy. And there you have a situation whereby mutations arise as a result of exposure to ultraviolet light. And so there you've got a lot of these so-called altered proteins that can be recognized by the immune system. And I think another interesting case is lung cancers, the lung cancers of smokers or ex-smokers. And those are lungs that have been subjected again to mutagens through cigarette smoke, actually respond better to immunotherapies than lung cancers that arise, in non-smokers. So it's not the whole story, because there are anomalies. So I wouldn't want to completely oversimplify the story, but there is certainly a skewing towards those cancers with high mutational burdens.
Really interesting. Yeah, I wouldn't have guessed that one. So what happens, well, not exactly what happens next, but when you have promising results, like Lorenzo was outlining there with the suppression of LAG3, how do you sort of take that forward towards therapies? I mean, let's assume that this idea really has something in it. What do you then do with that knowledge?
Yeah, Lorenzo, you might like to talk about the small molecule libraries.
Yeah, so when there's a promising target, for example, like LAG3, there's a variety of ways, as I mentioned, in which these we can then go on to develop treatments. And one of the ways we have been trying to develop treatments that are relatively more accessible is through doing small molecule library screenings. So these are sort of screening approaches where we order from companies or through collaborations with the School of Pharmacy, really large amounts of different drugs. And we then, in these, in a Petri dish experiments, test this really large range of potential drugs to see whether they, first of all, attach onto LAG3 and whether they then improve immune function. And then I think, when we have what we call hits eventually, so these are drugs that show promise in a dish, then it's a matter of eventually testing these in more complex models. And then, from there, you go on to eventually applying for, funding to undertake clinical trials, etc. So it's quite a long process.
Yeah, great. Thanks very much. So somebody's asking, what is the breakthrough needed? I don't know if there's an answer to this. What is the breakthrough needed for immunotherapy to be a widely effective cancer treatment, and how long might it take to discover this?
Yes, well, I think that understanding the precise nature of the targets that are recognized by the anti-cancer T cells is extremely important, as a breakthrough. So what happens here is we unleash an immune response by taking the brakes off, by blocking the stop signals, by removing the regulatory T cells. But actually, these are pretty blunt tools, because what we're doing is literally taking the brakes off the entire immune system. And then we're hoping that some of those T cells that are activated as a result of that will be cancer specific. And of course they are, in the cases where it works really, really well. Those patients who do well from immunotherapy have generated a T cell response that sees the cancer. But we still don't actually know what precisely that T cell is seeing. So what exactly is the nature of the target on the tumor cell? And there's a huge amount of work going into trying to identify the relevant targets of cancer-specific T cells. And I think therein will lie an extremely important clue.
It's just all about, isn't it, understanding fundamental mechanisms, and that's extremely difficult despite everything that we know at this stage.
Yeah. And actually, there's a lot of that going on in Cardiff. So there's a lab at the group of Andy Sewell, based at the School of Medicine. What they actually do is they work with T cells that are isolated from a successful immunotherapy. So they have so-called tumor-infiltrating lymphocyte products that they then use to fish out targets. So, that's an approach that tries to get to the heart of this using so-called successful T cells to start off with. They're the sort of bait to pick up these targets. So there's a lot of effort going into that.
It makes a lot of sense. Finding ways to be more discriminating rather than just throwing the pebbles at a wall sort of thing.
Absolutely. But, then you've got other bottlenecks as well. So very often when you look down a microscope at a cancer, what you can see are cancer cells, in a sort of, imagine a nest of cancer cells. And then you'll see all around them immune cells, almost forming rings around them. It's hard to think that those immune cells are not there for a reason, because they've been attracted to the cancer, yet they can't penetrate it. So there's also issues of access. Cancers do things like they deposit sticky matrix proteins, we call it. So these are thick, gloopy proteins that sort of form in fibers around cancer cells, almost forming physical barriers. So we need to also try and find ways of breaking through these things. We've talked about making immune cells or engineering immune cells to produce digestive enzymes so they can actually eat their way into tumors. I get quite excited about this because I have that it's one of the projects that we're sort of running in our lab at the moment. So you could empower immune cells with the ability to not just kill the tumor, but also to find its way there in the first place. Blood vessel conditioning, that's another way of doing it, improving the condition of the blood vessels that feed tumors, because they're often very, very chaotic because they grow in response to lack of oxygen and conditions that are suboptimal. So the blood vessels don't function that well. And, that's also therefore not great for trying to get T cells to the right place at the right time.
Okay, that's great, thanks. So there's a nice question here. Excellent showcase, thank you, Awen and Lorenzo. If you could choose only one, and this is impossible again really, diet, exercise or genetic predisposition, which is the biggest threat to making all cancer survivable?
Lorenzo, do you want to take it because you had your slide? Or
can you repeat just the
yeah, it's a question of where, if you could only choose one of diet, exercise or genetic predisposition, which is the one of those that is most likely to make cancer survivable?
I don't know. I think that it probably again depends on the cancer. I mean, it's hard to give a specific answer. We know there are certain cancers, like certain breast cancers, that have a very specific genetic predisposition, and of course that's also now knowing this through the help of basic research, we are now able to take the accurate precautions in these patients to reduce the risks that they do develop these cancers. But I think also diet and having a healthy lifestyle is extremely important. It's hard for me to pick one.
And by and large, cancer is a disease of aging, that by and large. And therefore, if you can keep yourself fitter for longer, that has got to be a good thing. And those are things you can do something about. So
improving cancers with the things you can control yourself. Nobody makes you put certain substances in your mouth. So yeah, there's quite a few more. We haven't really got time for all of them, but there's an interesting one about what are your general thoughts on the characterization of cancer immunotherapy by the media? Does it tend to get misrepresented? I mean, there are media outlets that divide the whole world apparently, substances that cause cancer and those that prevent them. What do you think about that?
Well, there's always, as a scientist, the feeling that any success story is rather overhyped. So I would always exercise a little bit of caution there. And cancer immunotherapy certainly has been subject to that. And I think, my first slide with the headlines from the Daily Mail, cancer immunotherapy, the great new hope, that epitomizes this sort of this issue. So on the one hand, I would say, that actually, one thing I would say is that, we have been talking about cancer immunotherapy. Well, not we have, but people have been talking about cancer immunotherapy for over 100 years. It's not a new thing. And, the National Cancer Institute in the United States was set up from the legacy of a guy called William Coley, who discovered toxins that could induce immune responses that could clear cancers. And this was, 100 years ago. And so what has happened in recent years that perhaps is worthy of a lot of attention is the understanding of molecular mechanisms and the molecular identities of these stop and go signals. And so from a scientific point of view, I can share the real excitement that brings to the table. So, I have certain sympathy in that way with the great headlines, because we have made undoubtedly transformational leaps forward, but it's still only, we've only sort of opened the tap to a certain extent, not to the extent perhaps that is, discussed in the media.
And there's not, this is kind of paraphrasing and answering one of the questions, which I think is right. There's not going to be a panacea that will just cure all cancer. It's always going to be, at least as far as we can see, pretty specific, even to patients. But there's just one last one. We've only got a minute, so you have to be quick with this one. Do you believe that the swiftness of the COVID-19 vaccine development could also have a positive effect on fast-tracking cancer-related clinical trials?
Absolutely. I mean, the COVID vaccines that, the Pfizer vaccines, the ones produced by BioNTech, were actually conceived from many, many years of cancer vaccine research. So, those mRNA vaccines were developed in order to vaccinate patients with cancer. And so a huge amount of effort has gone into optimizing the formulations that produce those vaccines. And when the pandemic came along, they made the switch to using their technology for COVID vaccines. But of course, this phenomenal, the cancer vaccine stuff never stopped, and this phenomenal, sort of worldwide experiment that's gone on to look at the efficacy of those vaccines can now feed straight back in to learning more about how to induce anti-cancer responses. So absolutely, yes.
Yeah, we've learned a lot, haven't we, about what is possible. But unfortunately, we're out of time. It goes by so quickly. But thank you both very much, talking about Awen and Lorenzo, for joining us and talking about these amazing and exciting research activities. And thank you, the audience, for joining us this evening. I hope you've enjoyed it. As I said, if we didn't get a chance to answer your questions specifically, we will follow up with you about that, and everybody will receive an email in the next few days with a copy of the recording and links to register to our forthcoming events. The next one in this series is on Thursday, the 30th of June. It's going to be Cancer Again, where we'll hear about current research into better and more accurate diagnosis in the case of prostate cancer. So we hope we can join you for that. If any of you watching would like to get involved with Cardiff University, volunteering time to help our students or helping us to fundraise for our research, please follow the link in the comments box for you. But that's it for now. Diolch a hwyl fawr. Thank you and goodbye, everybody. Bye.